Greetings and welcome to all the learners for the topic DNA footprinting. Why to learn DNA footprinting? It becomes necessary to learn the DNA footprinting assay as we all know that gene regulation is crucial for cellular function and differentiation, and this assay helps us to learn about the interactions or binding of transcription factors or regulatory proteins like promoters, enhancers or silencers which could enhance or repress the transcription of individual genes within the genome. So defining the technique, we can say that DNA footprinting is a method used to identify the specific binding sites of proteins on DNA. By analyzing the patterns of protected and unprotected regions, researchers can determine how transcription factors interact with the DNA, providing a profound insight of gene regulation mechanisms as it can be used for assessing transcription factors which bind to promoter, silencer or enhancer region of gene to regulate its expression. Principle of the technique says that DNA footprinting assay can be simplified as a technique of protection of sequences that are bound to specific proteins, that is the transcription factor or the regulatory proteins, which keeps the bound DNA sequences unaffected by the digestive action of any cleavage agents like DNases or hydroxyl radicals. The steps involved in DNA footprinting includes amplification the gene or the region of interest, labeling of the DNA with radio or fluor label, incubation of DNA with protein and immunoprecipitation, subjecting the amplified DNA to digestion by cleaving agents which can be enzymes like DNase 1 or exonuclease 3 or non-enzymes like hydroxyl radicals and UV irradiation. Gel electrophoresis of DNA fragments, results interpretation. Amplification is the first step in which the interested region or gene which is expected to contain specific sequences eligible to act as binding site of ligand or protein. Amplification is done using polymerase chain reaction technique to yield multiple copies of the gene for assay. Labeling involves either the radio labeling technique or fluorescence labeling technique. 3' or 5' end of the DNA is labeled using radioisotopes or fluorescent molecules. The next step is treating the labeled DNAs with the protein, which is called as inoculation with protein and immunoprecipitation. To assess if the protein has a binding site or not, the DNA is incubated with protein solution. Immunoprecipitation is achieved by the protein-compatible antibodies which precipitates the protein-bound DNA, and the free DNA is removed by washing. The next step in the procedure is digesting the DNA sample using cleaving agents which could be DNase 1 or UV irradiation or hydroxyl radicals. Learning in detail about the digesting agents, the most widely used is DNase 1, which binds to the minor groove of the DNA and breaks the phosphodiester bonds and separates the DNA. It works on many sites and produces many fragments of DNA, which makes it the most effective nuclease or DNA modifier. Other than this, another major benefit of using DNase 1 is that it can be controlled by adding EDTA to stop the reaction. UV irradiation can be used to excite nucleic acids and create photoreactions which results in damaged bases in the DNA strand. Photoreactions can include single strand breaks, interactions between or within DNA strands, reactions with solvents or cross links with proteins. The next is the hydroxy radicals. Hydroxy radicals are created from the Fenton reaction, which involves reducing Fe2 plus with H2O2 to form free hydroxyl molecules. These hydroxyl molecules react with the DNA backbone, resulting in a break. Due to their smaller size, The resulting DNA footprint has high resolution. Unlike DNase 1, they have no sequence dependence and result in a much more evenly distributed ladder. The negative aspect of using hydroxyl radicals is that they are more time consuming to use due to slower reaction and digestion time. Analysis by page and interpretation of results. The digested fragment of protected and unprotected DNA is subjected to polyacrylamide gel electrophoresis along with a standard sized DNA ladder. At the end of the run, a differential banding pattern is observed. The results are observed using autoradiography, traditionally or through elimination, showing a gap footprint of DNA in protein incubated DNA samples showing a specific binding site to the specific regulatory protein. The picture summarizes the DNA footprinting technique, which shows the experimental sets with one set of radio-labeled DNA untreated and second set with radio-labeled DNA treated with protein. When both are subjected to DNA's treatment, it results in different banding patterns. Upon visualization, we can see a gap in the protein-treated DNA banding pattern, which confirms that the binding proteins has left the footprints of them on the sequences, confirming the presence of specific binding sites on the interested gene fragments. Thank you.